Hello, and welcome to another virtual field trip to the New York State Museum. I'm Dr. Lisa Amati. I am the curator of paleontology at the New York State Museum and also the state paleontologist of New York. Now, a few weeks ago, we talked about some interesting invertebrate animals. So let me review just for a minute about what an invertebrate is. First of all, vertebrate animals are animals with vertebrae or a backbone. They include things like fish, salamanders, dogs and cats, turtles, dinosaurs, and even people. We relate well to vertebrate animals because, well, we are vertebrate animals, so we understand them a little bit better. Invertebrate animals have no backbone, they have no vertebrae. They are things like clams, snails, insects, lobsters, animals that are a little bit harder for us to relate to because they're different from us. But because they're different, they actually can be quite fascinating. They can do a bunch of things that vertebrates can't do. So before we get started, let's look very quickly at a geologic time scale. We looked at it last time too, and what you can see is time from when the Earth first formed and up through when first organisms evolved. They're all single-celled organisms, all the way up to here, the Phanerozoic. And this is when we have animals that are visible, large enough to see without a magnifying glass. And we can take the Phanerozoic here and expand it out so that it's a little bit larger. We see that there are three time periods: the Paleozoic, the Mesozoic, and the Cenozoic. Most of the animals that we're looking at today are still alive, but most of them that are fossils that we're going to look at are from the Paleozoic period. So we're going to be looking back in time to understand more about modern animals by looking at their fossil relatives. And we're going to start with the cephalopods. Cephalopods are animals like this cuttlefish, which is a highly intelligent animal. They swim around and they can change colors. They also include things like this squid toy that I'm holding. This is a toy of the giant squid. And they include the octopuses. Here's a giant Pacific octopus. Now, these animals are one of the most interesting types of animals to me because they are the most intelligent of the invertebrates. This is something that I would like to study if I studied modern animals because they're really smart. Octopuses can open jars, they can solve mazes, do all sorts of interesting things, and they can fit through tiny areas. Now, cephalopods have a wide variety of different types of shells. The cuttlefish that I showed you a minute ago has a shell that looks like this, and it would fit inside the body to help stiffen the body for swimming better. If we look at a squid, like the one in my arms, they also have a small shell, very lightweight, that runs down the back. But they're very fragile and they don't preserve very often. If we go to the octopus, these can fit through tiny areas because they have no shell left at all. So they're unshelled animals that can fit through teeny tiny little spaces. Now, there is one modern cephalopod that still has a hard outer shell, and it looks like this. This is called the chambered nautilus. And of course, I have a toy of a chambered nautilus to show what they would look like when they were alive. These guys have a big, heavy outer shell that makes them swim very slowly, and they're pretty clumsy. So what happened over time was cephalopods back in the Paleozoic mostly had these big hard shells, but they swam very slowly. So over time, they've lost their shells, like the octopus, or the shells have been reduced in size to make them swim faster. Now, the way that they swim is they have this little tube right here. It's called a funnel or a hyponome. Let me move the arms so you can see it. And they can suck water in through that tube into an empty space in the body called the mantle cavity. They do that by expanding the muscles around the mantle cavity to suck water in through the tube. Then what they can do is they can squeeze the muscles around the mantle cavity, shoot water out of the funnel, and fly through the air at a really high rate of speed. In fact, not only are cephalopods the smartest invertebrates, they're also the fastest marine invertebrates, moving at about 36 kilometers per hour, which is 22 miles per hour. The Japanese flying squid is the fastest invertebrate. So that's a little bit about cephalopods, but let's look at what they looked like back in the Paleozoic. So here's one here that is coiled like the chambered nautilus. There's a little bit broken off on one side. And then the tentacles would have come out from this area right here. So we can compare it to this toy of a Paleozoic cephalopod, and we can see the coiled shell and where the tentacles stick out. Now, not all of them were coiled in the Paleozoic. Some of them, like this one right here, had a straight shell, and the tentacles would have come out the front. Now, these look very clumsy, and it was a little bit difficult for them to swim. So they have a special feature that helps them to move upwards in the water column or sink lower down into the depths. Most of the animal lives only in the front part of the shell. The back part of the shell is mostly empty, and this one has been cut in half so that you can see that most of the shell, especially back here, was empty. Now it's been filled in with a few minerals. And there's this tube that runs down the center of the shell called the siphuncle. The siphuncle can actually release gas into these empty chambers or fill the empty chambers with water. If they put gas into the chambers, it would make them float higher up in the water, and if they put liquid into the chambers, they would sink lower down. So cephalopods live at a variety of depths in the oceans. The modern ones can mostly live down deep at night, and then some of them will go up towards the surface to feed. The ancient cephalopods also lived at a variety of depths and are really important because early in the Paleozoic, they were the top predators. They probably could eat just about any type of organism they wanted, and they would do this by catching the organism in their tentacles and then ripping into them with this sharp beak that's in the middle. We also know that the Paleozoic cephalopods had that beak. So they were super important predators, well, until fish evolved jaws. Now, when that happened, the fish were able to bite into these hard shells of the cephalopods, and it would make them sink. So a new type of cephalopod evolved. This one's kind of little, but you can see that the coiled shell is very complex. And this helped them to withstand bites from things like fish and still be able to float. Well, over time, these big heavy shells just became too difficult to manage, and now most modern cephalopods don't have the hard outer shell except for the modern chambered nautilus. So that's some information about cephalopods, which are one of my favorites. The next group of animals we're going to look at is the bivalves. We can look at this set of bivalves from the Paleozoic. These are Devonian in age, called Gramecia. And you can see that they're made up of two sides to the shell, and that gives them their name bivalves. They have two valves to the shell. Bivalves are extremely important because they can live in a wide range of environments. They can live anywhere from freshwater to saltwater and everything in between. So they can live in places where other animals can't survive. And they're an important source of food for lots of different kinds of animals, including us. Bivalves hold their shells shut by using muscles, and we can use this scallop as an example. So when the shell is closed, it's because they have muscles that are holding the shell shut. When they want to open the shell, they relax the muscles, and a rubber band-like ligament at the hinge area will allow the clam to open. Now, this is why you shouldn't eat clams from the grocery store that have the valves open, because that means that they've probably died and the muscles relaxed to open the valves. Clams, or bivalves, live in a wide variety of environments, as I mentioned, and they also have a lot of different shapes. So let's look at some different types. Here we have oysters. Oysters usually live by cementing themselves to something hard like a rock. So this is the bottom shell of the oyster, and this is the little top shell that would flip open. And oysters will also cement themselves to other oysters. So here's one cemented to somebody else. They can be free living like this giant clam right here. These are also known as man-eating clams, and that's because they have a symbiotic relationship with photosynthetic algae. And the algae help to provide them with food so that they can grow really big. Razor clams, like these, live burrowed into the sediment. And if you step on them, they can cut your foot, which is why they're called razor clams. Then we have the scallops, like this one. And I'll show you using the scallop toy how they're able to move. Basically what they do is they clap the valves together really fast, and water shoots out the back, and they'll kind of flap through the water to escape a predator and flap through the water to escape a predator. So it's not a super efficient way of moving, but it works for them. So let me show you how bivalves eat. They're important organisms to help with cleaning the water. So what they do is they have this little red inhalant siphon, and that's where water is drawn into the shell. It goes over this kind of striped thing right here, which is the gills, and carbon dioxide is exchanged for oxygen. Then the water is circulated back out through the green exhalant siphon. Now, while that water is circulating over the gills, little tiny food particles are grabbed and passed towards the mouth, that tiny little dot right there. So that's basically all they have of a head, is that mouth. And if they're lucky, this little red thing right here, which is called a palp, and the palp can be used to help shovel food into the mouth. So their head is basically a mouth and two food flaps. So they may not be the most intelligent of animals, but they certainly are delicious. Next on our menu of delicious invertebrates, we have the snails, or gastropods. We have a number of fossils here for you to look at while I tell you about gastropods. Gastro means stomach, like a gastroenterologist, and pod means foot. And they get the name because the big fleshy foot of the snail looks like it's where the stomach would be. So that's why they're called that. Gastropods can live in almost any environment. They can live on dry land, they can live in fresh water, they can live in salty water. In fact, they can actually live in water that changes from being fresh to salty, like in an estuary. And they can do this because they're osmoconformers, or osmoregulators. And it means that they can adapt to changes in the amount of salt in the water while they're alive during their life cycle. So they can live in places that other animals just can't live. Now I'm going to tell you about how gastropods eat. They eat using a special mouth part called the radula. And the radula is basically a tongue-like strap that's covered in these sharp scales that help to scrape food from the rocks. So let's look at a picture of the radula. There's a cute little snail. The middle picture shows the radula here. And the brown spot is muscles that control the radula. So what they can do is they can stick it out of the mouth and use the sharp scales to scrape algae from rocks or shells or something like that. And I'll show you a close-up of the radula scales. This is a super zoomed in close-up using a scanning electron microscope. And you can see how the radula would be very sharp and able to scrape even hard algae from rocks. Well, they can also use the radula in another way. Basically what they do is there's a certain type of snail, and they've been doing this for 465 million years. There's one type of snail, here's an example of one right here, and they have a big, huge, gross, fleshy foot. What they do is they move onto the top of something like this bivalve right here, and they use a special radula to drill a hole through the shell of the bivalve. And let me move this snail out of the way so you can see the hole a little bit better. The hole is usually a slightly larger on the outside than on the inside, and that's how you tell that it was drilled by a snail. Now, okay, so the snail has drilled the hole, and now what's it going to do? I mean, it doesn't have any arms or legs or anything. How is it going to get the clam out of the clam shell? What they do is they take their tube-like mouth, they stick it through the hole, and they basically throw up inside the clam, and they digest the clam, and then they can suck the meat out of the clam into their mouth. So it's really gross, but it's also super cool because, you know, who knew that a snail could actually kill and eat other animals? Now, it turns out that there's a snail that can actually kill people. And can you imagine how embarrassing that would be? No, it wasn't a crocodile, it was a snail. So let me show you these cone snails here. This is called conus. The shells are collected by people who like what they look like. But the snail is actually interesting because when it was alive, it had a very special type of radula that looked like a harpoon. So here's the radula here. It has a very sharp tip and it's hollow. And what the snail does is it oozes up next to something it wants to eat, like maybe a fish or something. And you know, the fish is unconcerned because it's just a snail, right? And it gets closer and closer, and then it shoots the harpoon out of the mouth, and it shoots so fast you can hardly see it even in slow motion video. And it shoots the harpoon into the fish, and then it ejects this venom into the fish that paralyzes the fish, and then as it reels the harpoon in, the mouth gets really big and just kind of engulfs the fish and takes it into its body and digests it. Now, these can be dangerous to humans because the paralytic that's in their venom can actually make your heart stop beating. But because we're so much bigger than the cone snails, they don't normally inject us with their harpoon because we're just too big for them to eat. So we're not prey to them. In any case, I wouldn't pick one up. Now we're moving on to echinoderms. And the first one we're going to look at is Seastars. Seastars are beautiful organisms that have changed very little over time. So this one is called Devon-aster eukarya, and you can see the beautiful sea star right here that's actually preserved with a bivalve. Here are the two valves of the bivalve. And if you compare that to this sea star that I bought at a gift shop near the ocean, you'll see that they're very, very similar. Seastars are echinoderms, and echinoderms move themselves by using these special little things called tube feet. Tube feet are super cool. They're basically little tubes with a suction cup on the end, and the little feet suction cup to the seafloor and can pull the animal around and over the seafloor, around objects. Let's look at a close-up of some tube feet on a sea star. You can see those little light-colored things with a suction cup on the end are the tube feet. And then this little red spot right here is an eye spot. And the eye spot is sensitive to changes in light and can also make out patterns and shapes. So in a way they can see, and they're definitely very good at moving themselves around on their tube feet. Now, sea stars, although they just look pretty, are actually also very serious predators. So you look at a sea star and you're like, well, how's that going to eat something? One of their favorite things to eat is clams. And the way they do it is they wrap their arms around the clam, attach their tube feet to each valve of the clam, and then pull the clam shell open. All right, well, now what are you going to do? You got the clam shell open. How are you getting the clam out of there? Well, this is super gross and also really cool. Basically, what they do is the mouth is in the middle part of the body, and they have two stomachs, an upper stomach and a lower stomach. And they take the upper stomach and they turn it inside out the mouth, release digestive juices, start to digest the clam in its own shell, just like the snail did, and then wrap the stomach around the meaty goodness of the clam, pull the stomach back up through the mouth, and then move the food up into the upper stomach for further digestion. So mostly they eat bivalves, but some types of sea stars can eat coral polyps. and they evert their stomach onto the coral polyps and digest them. If the population of sea stars gets high enough, they can actually devastate entire parts of coral reefs. Let's look at another type of echinoderm. This time, it's sea lilies or crinoids. They get their name because they look kind of like plants, but these are animals related to the sea stars. Most of the body is in this cup-like calyx, and then they have arms that branch up off the top, and they're lined with tube feet. And the tube feet grab food particles and pass it down to the mouth in the calyx. They're raised above the seafloor on this stem, and many of them have a holdfast at the bottom to attach themselves to the seafloor. Now, if you look at the holdfast, the stem, and all the way up to the arms, you'll see they're made of these individual little pieces or elements. Those little pieces or elements are hollow in the inside, and they kind of look like Cheerios or little buttons, and the flesh of the animal runs down through those elements. Now, the cool thing about this is if you stack a bunch of Cheerios on top of each other and hold them together with a membrane instead of fusing them together, it allows the stem of the crinoid to move. The arms can wiggle up in the water, and it gives them more freedom of motion. Now, unfortunately, that also means that when the crinoid dies and the membrane rots, all of the little pieces fall apart. So these are tiny little stem pieces of crinoids that just fell apart when the crinoids died and the waves crashed over them. We also have a fossil here showing the holdfast. And if you look at the very center of the holdfast, you can see a star-shaped hole. Those are the holes in the middle of the elements that run through, or that make up the stem. And the body of the animal runs through that little hole. Next is a crinoid that's almost complete. It doesn't have a holdfast at the bottom. It has fallen off. You can see all of these little Cheerios stacked up to make the stem. There's the cup-shaped calyx. And here are the arms reaching up into the water to grab food particles. We have another one here that shows the calyx and the arms a little bit more closely. You can see that the calyx is made up of a bunch of little pieces that are fused together. There are the little Cheerios that make up the arms, and this one has branches off the arms, also made up of little teeny tiny Cheerios. Now you can imagine that living attached to the seafloor, that these animals would be easy to prey on. So when fish evolved that could bite into hard objects, they would prey on these crinoids, and the crinoids are just waving in the ocean currents. They can't escape. So over time, crinoids many of them have lost the stalk and consist only of the holdfast at the bottom, a little tiny calyx where most of the body is, and huge long arms that they can wave in a coordinated motion to swim through the water and escape predators. So in the Paleozoic, these guys were so common that they actually could make thickets of crinoids that were extensive over a wide area. And it was kind of like they made their own little habitat. So there are tons of these sea lilies or crinoids living together, and they made a habitat for other organisms to live in, increasing diversity of the different types of animals on the seafloor. So crinoids were really important in the Paleozoic. They're still alive today, but they don't really look very similar anymore. Our last group of organisms today is the Graptolites. Graptolites were a really important type of plankton in the Paleozoic. And plankton usually refers to organisms that are relatively small and float in the water column and are carried wherever ocean currents take them. Graptolites usually lived in the open ocean, floating near the surface. So when they died, they would sink down into the depths of the ocean where they would be preserved. Now, normally these deep rocks are dark in color, like this black shale, and the graptolites are preserved as a thin film of carbon on the shale, making them a little bit difficult to see. In fact, it kind of looks like somebody was walking over a bunch of dark rocks with a pencil in their hand, tripped and fell and drew a scribble on the rock. Now, it's funny that I should mention that because their name is actually Graptolite. Graptos means written and light means rock. So these are written rocks. That's how they got their name. Graptolites could live in a wide range of environments, mostly deep though. So most of them didn't live very close to shore. The good thing about them living in deep water is that they tended to live all across the globe, and they evolved relatively rapidly. This makes them excellent index fossils in the Paleozoic. And what that means is that we could find something like these graptolites here. This is a monograptid. Here we have a dendroid graptolite. And then two samples of a very special type of graptolite called Climacograptis bicornis. Now, Climacograptis bicornis only lived for a short period of time, but it lived all across the globe. What's cool about that is that these come from New York, and we can identify them as Climacograptis bicornis, then compare them to the exact same species found somewhere else in the world, like in Great Britain, and we know that the rocks are the same age because they only lived for a short period of time. So it allows us to date rocks that are found in very different parts of the world. Graptolites are colonial organisms, and this is an example of what one of the colonies might look like. The colony has a bunch of little cups on the side of it, and one individual graptolite would have lived in each one of those cups. So let's look at another picture. This one is cutting away the side of the colony so that we can look on the inside and see the little organism, which has been colored purple here. So the graptolite is a very small animal, very simple. It has two branches that stick out of the opening in the cup on the side of the colony, and they grab little food particles out of the water. Now, each one of these little animals would have been attached to the animals living in the other cups of the colony. And that's neat because that means that if something happens to somebody on one side of the colony, the individuals on the other side of the colony are aware of what's going on. So graptolites are really important for biostratigraphy, but they're also important or also interesting animals. So those are all the animals that we have to cover for today. I hope you'll join us for another episode of the virtual New York State Museum. And the next time we'll be back, I'll be talking about trace fossils. Thanks for coming, everybody.